Here we go. It's officially day 2 now. All right, so I'm going to start off on the side of the day 1 review. The day 2 review sheet, the side that has your name space left on there. And of course, we're going to be hitting up each of these boxes individually. I'm going to try to save ourselves a little bit of headache here in the future. And also, we're going to distinguish everything. You still have spaces to write down questions, so as you think of those, stop me, or ask them whenever's most convenient for you. Remember, I'm here after school too. You guys ready? Let's do this thing. Types of cells. Okay, so types of cells, we've got two types of cells, two major categories. I affectionately call them prokaryotes and eukaryotes. Prokaryotes and eukaryotes are two distinctive types of cells. They are the two main categories. Even when we go back to talking about domains, from like domains and kingdoms and classes and orders and phylums and all that stuff, we basically distinguish living things in the categories of prokaryotes and eukaryotes, and there's a small distinction between eukaryotes. Anyway, so prokaryotes, what's their main difference from a eukaryote? Yes, sir. They are big dummies. I like that. They're actually rather small, but what do you want to give me in terms of detail?
They don't have a membrane around the nucleus.
That's true. So I'm going to focus on one thing you said there. The big distinction here is that these have no nucleus. I'm even going to put a star next to this. It's so darn important because that's literally how we name these two types of cells, based on the presence of a nucleus, or for the case of a eukaryote, they have a nucleus. Okay? That's a huge distinction. And even the phrase or the term eukaryote means that it is good kernel. And a good kernel, of course, is going to mean that it has like a good center, and it has a little control center, and it's going to be the nucleus. So prokaryotes also lack something else. So they're mostly known for what they lack. What else do they lack? What do you think?
Membrane bound.
There we go. That's it. Membrane bound organelles. Okay? So they have no membrane bound organelles, whereas for eukaryotes, they do have these. Okay? That's one of the main distinctions between them, because whenever you look on the inside of a prokaryotic cell, you're not going to find a lot. There's not a lot of cool stuff. You're going to find their DNA. It's not contained in a nucleus. It's just kind of randomly floating around. They don't have cool little specialized parts and pieces, so they don't have these membrane-bound organelles. Now, I also just want to make a small note here. This does mean they still have ribosomes, because they still got to make proteins. That's really important to them. And there's actually not a specialized membrane on a ribosome, so they would still have that. What else do we know that's different about prokaryotes and stuff like that? They could, yeah, in a lot of cases they could. Not always, but they could. But I don't think that's distinctive enough for us to write it down. But that's a good point, a good thing to note. How about this? How many make up a living thing for a prokaryote? Yeah, so we usually use the phrase unicellular to describe a prokaryotic cell. There are some very, very rare exceptions to this, but eukaryotes are either multicellular or they're unicellular. But the cool thing is that they can form more complex structures and become multicellular. What else do we know that's different between the two? Okay. Overall, they have low complexity over here. And then eukaryotes have higher complexity. And I'm also just going to go and distinguish eukaryotic cells can become specialized because they have specialized organelles, and these are not or non-specialized. Prokaryotes, almost all of them seem to operate basically the same way. Yeah, they might have different kinds of little tail things that help them move around differently, but they basically operate and perform cell functions the same way. Whereas eukaryotes, there's a lot of distinctions there. Some cells have a lot more lysosomes or peroxisomes that help break things down. Some cells have larger vacuoles. Some cells are flatter and wider, so they have a particular purpose when it comes to a multicellular organism like skin. So they can be very, very, very purpose-driven, and that would explain why their form is the way that it is. But prokaryotes and eukaryotes, basically, you carry eukaryotic cells all around, right? You carry them around because you're made up of eukaryotic cells. You're a complex organism. Whereas prokaryotes, no, you don't really do anything with the, well, actually, you do a lot of things with them, but they don't make you up. So that's the main distinction. How you guys doing? Good? Let's go down. Let's do plant cells and animal cells. How's that sound? Yep, it clicks. It clicks. Now it happens. So plant cells and animal cells, they also have some differences. What's the difference between a plant cell and an animal cell? Yes, sir? You got it. That's one big distinction. Plants definitely have a cell wall. What else?
Large central vacuole.
Wow. Big phrases. Large central vacuole. Okay. What else? They do have chloroplasts. Anything else? Well, I think that greenness would be caused by the chloroplast, which contains this thing called chlorophyll. Good call. Anything else that really distinguishes them? Yeah.
Aren't they grid-like?
I guess they could organize themselves in a more grid-like pattern, but we still have different kinds of tissues found in plants, so it'd be a little hard to get away with saying. So we feel like we're tapped out there? Okay. Well, then let's go ahead and talk about animals. What makes them so distinctive? Yeah.
They only have mitochondria.
Oh, interesting distinction here. Only mitochondria. And that distinction is important because that says animals only have mitochondria, but that also implies that plants have chloroplasts and mitochondria, which is really important because plants, while they make sugars from chloroplasts, they got to break down those sugars to get energy. So they have to have their own form of cell respiration and their own organelles to do it, which are mitochondria. What else do we know that's distinctive for animal cells? Yeah.
They have like a bunch of little vacuoles.
Smaller vacuoles. And that's probably because animal cells probably ingest a lot more variety of things, and they want to isolate those reactants from each other in a little bit more safe way, instead of having one big thing that swells up with water. Yes, sir?
They don't have a cell wall.
No cell wall. Which also kind of implies, using the phrasing that you used earlier, they only have a cell membrane, right? And this is an important distinction just because we know animals got to have a barrier, and that cell membrane acts as the barrier. Plants still have a cell membrane, but they also have a cell wall, like an enhanced layer, because that's basically where their structure comes from. Any other major distinctions we care about? Yes? There's still cytoskeleton in the plant. It's just not as prominent for support, but it is used for some other stuff. You will find more of them there, but I wouldn't say only. I think we're pretty much tapped out here. So let's go ahead and hit up these pictures. Hey, look at me. I don't have to draw them. Isn't that great? Aren't you proud of me? I know you are.
I wouldn't want to see it.
I wouldn't either. So we want to go ahead and label these things as best as we can. Let's do a little green for the plant cell here. Hey, look. It's a plant. So A, what part is A pointing to? It's pointing to the vacuole. What about B? It is the cell wall. C? The rough ER. D is pointing to the nucleus. What about E? I think I heard it, yeah?
Powerhouse itself.
I'm writing mitochondrion because it's only pointing to one of them, but if there's plural, you'd say mitochondria. F, what are we pointing to? Yeah, chloroplast. And then G. Yeah, I'm gonna agree with you in this capacity here. I'm actually gonna agree with Smooth ER on this one. The reason for this is because it's supposed to look a lot like this, and it's not directly connected, which is gonna be okay-ish. But I think we'll be able to distinguish it a little bit better on the next picture. But I can see the confusion, and once again, I didn't make these pictures, so maybe that was the original intention of this artist, but I don't really care at this moment. I'm just trying to like look at things and see what they are. Anyway, so I'm going to change colors. I don't know why we always use the color red for animal cells, but I always manage to. So A, what's that? Nucleus. B, what's that pointing to? Well, it's this line here.
Oh, I thought it went all the way through.
No. I'm going to agree on this one. Now, once again, I'm probably making this artist very upset for using their stuff, but hey, public domain. But also, I'm going to label G as the Golgi apparatus here, because I think this green portion should be the same thing. It just shows how common it is and how important probably protein production is in animal cells. C, what's that pointing to? I'm going to agree. It should be the nucleolus. D is pointing to some interesting stuff here. I know you may have a hard time with this, but this should be pointing to a type of the cytoskeleton. Those little lines there represent some fibers. Those are protein structures. E is pointing to cell membrane. F is pointing to, it is, I know that pretty easily because it's got little dots all over it, but also it's really pointing to this structure that's connected directly to the nuclear envelope, which gives it away. H, I know it's a little hard to tell, especially in your paper.
Mitochondria.
Not quite.
What?
Yep. Yeah, it is a lysosome. Because they're kind of small, they're filled with enzymes, and they're going to break things down for us. But I... Yeah. And then J, I know it's hard to tell. It looks like a little like scratches on the side. This is also the cytoskeleton. In this case, it's a different type of portion of the cytoskeleton. And then we have K. Yeah, so you'll see that we have like a smooth ER here, and I think this is kind of the same thing. So we're kind of treating this the same way we did with the green stuff, but this should be the smooth ER. Okay. Do you guys have any major questions there, concerns, issues, anything you want me to address, point out, help you identify? No?
Why are there two Golgi apparatus?
Because that's probably, and I'd say there's 3 here, and the purpose for that is, my guess is because they're showing or implying that protein production is really, really, really important for animal cells, which is true. They make a lot of proteins, and they got to sort and organize a variety of those things, so that's probably what that is. The smooth ER, it's for lipid production, but hey, I'm kind of giving things away because, papa, we're talking about the cell organelles. Here we go. I'm changing colors.
I remember this test like it was yesterday.
Well, for you it was. Because somehow we've traveled back to the past. Okay, so, nucleus. What's it do?
It's the big brain.
I wouldn't quite say the brain. It does an actual thing.
It makes
Okay, yeah, so it contains the DNA. That's its job. That's its distinction. It holds the DNA. Notice, this doesn't suggest that it leaves the nucleus. The DNA always stays housed within the nucleus, and that's important. That'll play a role later when we talk about other processes. We've got the nucleolus. What's that doing? Yeah, actually, it's exactly what it does. It makes ribosomes. And ribosomes are really important to us because they do what? Now, proteins are kind of the workhorse of the body. So we got to have a lot of these made all the time. And ribosomes are little protein factories. Some are free-floating, some are stuck elsewhere. But that's the main job, is they're making a ton of proteins all the time. So we got ribosomes. What about the cell membrane? Sure, yeah, I agree. Anything else it does? What do you think? What did he say? Okay. What did you?
He said this regulates what comes in and out.
Actually, that's exactly what I'm looking for. So I'm gonna say it's kind of a, let's say it's a protective barrier, and we're gonna be used. It's mostly used for transport of things in and out of cells. That's basically what you said, right? It regulates, and that's its function. That's very good to distinguish there. Good job. Cell wall. What is it doing? Yes, sir. It is found in only plants, I agree, and it is kind of protective, but also it does. Right, so it gives physical structure to cells. Remember, that's only plant cells and fungi cells, but it gives physical supporting structure. Mitochondria. Yeah, it's the powerhouse of the cell, but also it should, which is the most common energy form, which we'll talk more about later. But basically creates energy from breaking, oh boy, down sugar, which we call cell respiration, by the way. I'm making a little note there to distinguish for you. How am I doing in terms of other things? I don't know. Vacuoles. Yeah, that's basically it. They store waste. In plants, they of course store water, which maybe isn't more so waste, but it's definitely a product. Lysosomes. Yeah, we'll say they're basically the recycling processor. These are really important because if we break down old parts within a cell because it's old, or maybe we need to distinguish things and say like, hey, I don't know, something like, like white blood cells. They use lysosomes to help release enzymes to help break down the bad guys that might be invading our cells too. We got the Golgi apparatus, which once again, you could call it the Golgi complex or the Golgi body. You could call it all kinds of things. I don't really care. But the Golgi apparatus, I told you when you see the word Golgi, you should think of FedEx, right? And so the Golgi apparatus does sorts and delivers proteins. Good call. Very important. Chloroplast. Convert light to sugar. And this process has a fun name. Photosynthesis. Rough ER. What do you think? I'll help you out, and I'll say it directs protein production. Because you're right. Basically, the ribosomes are stuck on the outside, and the ribosomes are making proteins. This helps direct them to the place that they need to go and holds those ribosomes. We've got the smooth, I almost wrote small, the smooth ER. This one might be a little tricky. Lipid production happens here. This is where all the lipids that go to make your cell membrane, as well as all the other vesicles and organelles, that's what makes their membranes up as well. That membrane-bound organelle is made basically here, starting here at least. Cytoplasm.
It's the jelly-like fluid that all the organelles are in.
It's the jelly-like interior of the cell. And I should also point out, it is used for some support stuff to kind of help, you know, the organelles stay a little bit more seated and stay where they're supposed to. But also, it's mostly water and mostly salt, and some processes happen here. If you guys remember talking about glycolysis, it actually happens within the cell cytoplasm. And cyto means cell, by the way, so it's the plasm inside of the cell. And then we have the cytoskeleton. I know there's a lot of components to this. There's really 3 main fibers that are involved with it, but it's a protein network. For structure and for transport. Namely, if you guys remember those cool proteins that kind of walk and carry vesicles around in the cells, they have like the funny shoes. Those guys, they're walking on the cytoskeleton. So they basically give like a little highway system for things to move around and follow. Make sense? Okay, do you guys have any questions on any of the cell organelles? No? Wow, look at us. We're doing pretty darn good. Like, we're doing pretty darn good. So I'm going to move on, and I'm going to talk about the cell membrane. And we're going to hit up as much of this stuff as humanly possible. We're doing this. I'm committed. So phospholipids, and I know your drawings look a lot better than what I'm going to attempt today, but that's basically a phospholipid, right? We know they have 2 main parts. We know they have a head, and we know they have a tail, or tails, right? We also know that this head has a special feature about it. It gets along with water, and water is a polar molecule, so we identify the head as being polar. Polar pop. And then we have a non-polar series of tails. Non-polar pop. So after this, we know that if the head can interact with water, it's because it prefers to. We give this a fun phrase. Hydrophilic. Yes, indeed. Hydrophilic. Phila, love. So it loves water. And the non-polar tails are hydrophobic. Water-fearing or water-hating. So knowing this, let's go ahead and look at that basic structure. And I'm not going to draw the whole thing because I'm super lazy. So when we look at this, this just kind of continues, right? Super lazy. So lazy, I can't even finish the word. You'll notice that there's 2 layers of phospholipids, right? This is what we call the phospholipid bilayer. Bi means 2, so there's 2 layers of phospholipids. Please take note of the direction that these are facing. They're facing in opposite directions because as a barrier, as a cell membrane, we're going to be able to see that the polar heads are facing outward, which means water can touch both sides of the membrane, but water can't really easily pass through. Now, some things can, but not everything can. We actually use this fun word called semi-permeable to describe how some things can pass through. Now there's some other parts of the cell membrane that we really care about. You might see some structures that look kind of like this. These are channel proteins. And of course, proteins are used to help move things in and out of the cells, and also basically can operate as like an anchor for some other things to attach to them. You might also see some other blobby looking things in here. In this case, those will be cholesterol. Not all cholesterol is bad. Yeah, some could become plaque in your arteries and stuff, but cholesterol itself is basically going to strengthen your cell membrane. That's its job. It makes it stronger. So we have a lot of lipids, we have a lot of proteins, we have cholesterol that's found in there, and there's even carbohydrates. If you guys look at one of the lipids, you might see a structure that looks like this. You guys see the ziggy-zag thing? Okay, those are carbs. And their job is basically to distinguish the cell from other kinds of cells. This is how cells communicate. They have little receptors on the outside, and they communicate and they transmit messages thanks to those carbohydrates. If you guys remember talking about antigens on, like, white blood, well, not white blood cells, but blood cells, that's basically an extension of a carbohydrate and some proteins. Anyway, yeah, so we've got those terms out of the way. Let's get the big idea out of here. Fluid mosaic. I'm trying to spell it phonetically. Mosaic model. The fluid mosaic model is basically we'll say mobile. And I'm really bad at spelling. I'm so sorry. The very mobile and dynamic membrane that is made of many distinctive parts. So we've talked about proteins, carbohydrates, lipids. They're all different kinds of macromolecules, but they're found in the same place. Another interesting fact is basically we didn't even used to think the cell membrane had that many proteins in it, but now we do. We think it's got a ton. The cool thing is the proteins move around too, unless they're like anchored to the cytoskeleton. They move around like a ship on the ocean, right? They're kind of stuck with the current, they're stuck with the waves, and they're going to go wherever that takes them. Sometimes these things flip over and they're changing all the time. This is cool. This is actually a lot more interesting and means basically proteins have a very important purpose elsewhere in the cell membrane too. There are three terms down here: hypertonic, hypotonic, and isotonic. I'm going to go ahead and start with isotonic because it makes the most sense to. So if we have a cell with a little nucleus in there, we have things moving out and we have things moving into the cell. We're going to think about this in terms of water and other things, which we call water the solvent, and we call the other things solutes. So we're going to say water, in this case, water is going to move in and out at equal rates for this cell, which also tells us we have an equal balance of salt in this case. It doesn't matter what it is. It could be sugar, it could be sodium, it could be chlorine, it could be potassium. But in this case, we're just going to say salt. It's even amounts, so we're just going to write little equal side off over here. They're equal rates of moving in and out as well. I'm just going to also make a note here. Animal cells, they love to be isotonic. It makes sure that there's an equal exchange and homeostasis is maintained. Now, hypotonic, you'll notice when I drew this cell, I made it much bigger. And that's because basically water keeps moving in, doesn't it? The reason for this is because salt concentrations in this case, or solute concentrations, are much higher than what they are. Woo! Thank you. Than what we find outside. And remember, every time you see the word salt, and I'm just doing this to give you a practical example, but we mean a solute, just any item that is something other than water. I'm also going to make a note here and say plants love to be hypotonic. The reason for that is because that means their vacuoles are swollen and full. That means their cell membrane, their cell walls are being pushed from the inside out, which means it's harder to compress them, which gives them more structural support, which is why trees can be so darn heavy, even though they're still made of cells with no bones. And then hypertonic, you'll see I've kind of drawn it all wrinkly.
Get out of the house.
Yeah, get out of here. Because I said if you're hyper, your parents tell you to go where? Outside. So outside is where the water is going to move, which causes that wrinkling or that crenation. which of course can kill the cell because it doesn't have enough water. This also means that we see that the salt concentration in this case is going to be really high outside of the cell and a really low concentration inside of the cell. I should also mention hypotonic for our cells, for animal cells, could be extremely dangerous because if they swell up with too much water, they can burst, which we call lysis. Question? Question? Any questions? Yes.
So the lock and key model has to do with like channel protein, right?
No, the lock and key has to do with enzymes. And enzymes basically help break down things. The other proteins that are like channel proteins, they just are open pipes that allow some things to pass through there, which helps make it semi-permeable.
The ones that let like certain things pass in, right?
Yes,
Okay.
absolutely. Okay, so can I move on? Soon. Can I move on?
Yes, sir.
Okay, so let's do transport. So when it comes to cell transport, we've got two different varieties. We've got passive and active. Passive means no energy is used, right? But active uses energy. So we like to use passive whenever we can because it's free. It just happens naturally. Thanks. So we have diffusion. Yeah, I tried to spell it. Osmosis. And then we have facilitated. So diffusion is the movement, oh boy, of a solute, like we mentioned salt earlier, that would be a solute, from a high concentration to a low concentration. Osmosis is the movement of water from high to low concentration. Facilitated diffusion is the use of a cell, let me try that again, of a protein in the membrane to cause diffusion. So if we have a pipe that only allows certain things through there, but it still moves from high to low concentration, that's facilitated diffusion. Water needs to pass through cells, but they can't normally because there's a nonpolar surface inside the membrane. So they have to use a channel. It's called an aquaporin. They have to use a channel to help transport water through there. That's a pretty big distinction, and it's still no energy. And then we have active transport. It comes in two varieties, endocytosis and exocytosis. Endo means into. So things are moving into the cell. Exo means things are exiting the cell. So things are leaving. Now endocytosis has pinocytosis and phagocytosis. Yeah, which literally means cell drinking. And cell drinking means it brings in a lot of smaller particles, whereas phagocytosis means the cell eats, or it's cell eating. This eats larger components, like larger blobs that are eventually absorbed into it. Phagocytosis is used in your immune system all the time because you have white blood cells that attack other living things. Pinocytosis is used usually for digestive purposes. How you guys doing?
Pretty good. How are we doing on time?
We're doing this. Moving on. Let me just remind you of our best friend. I'm just going to title this one Love. Because the triangle of love, yours is going to be a little bit better looking than this one, but it still loves you. It loves you. So the triangle of love. Oh gosh. Oh gosh. So basically we have high concentration over here and low concentration. This line, this slope that you see, that is called the concentration gradient. That's what this shows you. And it always helps you realize if it's active or passive. So basically if we go from high to low that we see here, this is going to be passive transport. But if we go from low to high, we call this active transport. Just like trying to ride a skateboard on a hill. If you just sit on the skateboard and go downhill, you didn't have to use energy. Gravity did it for you. If you're on a skateboard at the bottom of a hill, well, you're going to be pushing for a while. You're not going to be able to cruise on this thing at all. Also, try not to forget that phrase, concentration gradient. That's whenever they're talking about it, they're talking about this slope that's found there. So keep that in mind. Can I move on? Okay, so it'll never leave you, don't forget. So plant parts, we've got roots. They anchor the plant to the ground, but also absorb water and nutrients from the ground. Stems move water and products to or from the leaves. And of course the leaves, photosynthesis happens here. That's why leaves are usually larger, right? They're wide, they have a flat surface, they absorb light really well because that's where most of the chloroplasts are contained. That's also why trees' bark or stems might be brown and the leaves are green, because that's where the photosynthesis takes place. There was also this really terrible drawing, which I may draw better on here than I did on your paper.
Oh, wow.
Maybe. This opening that's found here, we call that a stoma. It's found on the bottom of leaves. Maybe the top side, but mostly the bottom of leaves. And this is where water vapor leaves. This is also how CO2 moves into the plant, and this is even where oxygen gas leaves. These are called guard cells. Well, okay, when it comes to plant parts, we have the male anatomy and the female anatomy. The only parts we super care about for males are going to be the stamen, which is going to be the male reproductive organ, of course, and that's just what we kind of call it. But there are two main parts that we care about. Those are going to be the anther and the filament. The anther is basically going to be the part that holds the pollen and produces the pollen, whereas the filament holds the anther further away from the body of the flower. These are usually really thin, kind of look like this, and usually have little ends like that. Whereas with the female reproductive system, of course, we call this whole thing the carpel, or sometimes it's called the pistil. But anyway, this is of course going to be the female reproductive organ. And then we're going to see three parts here. We're going to have the stigma, we're going to have the style, and we're going to have the ovary. The stigma, style, and ovary are kind of from top to bottom. This is going to be the portion that catches the pollen. And then down the style, we're going to see that this is where the pollen tube is. And this is just going to transport the pollen basically all the way to the ovary. And this is where fertilization occurs. So if we were to draw this, it would kind of look like the top portion like that. You got a long stalk almost, and then the ovary. Inside of here, you'll see little sections where basically seeds will develop, and of course, with a lot of fruits and vegetables, of course, this can swell up, and that will become, what you know as the fruit and vegetable. Okay, so that's everything for day 2.